Hello, everyone, and welcome to Precision and Efficiency Multiplexing in qPCR for Assay Development. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. We do encourage you to participate today by submitting your questions at any time you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You can also submit any technical issues there as well if you're having any trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Chia-An Yen, molecular biology scientist, Thermo Fisher Scientific. For a complete biography on our speaker, you can click on the biography window at the left of your screen. You can now begin your presentation.
All right. Hi, everyone. Thank you for joining me today. My name is Chia-An, and I work in R&D as part of the qPCR assay and contents development team in South San Francisco. Today, my goal really is to provide you with some guidance, insight into how to go about developing a qPCR multiplex workflow. And that hopefully by the end of this presentation, you'll feel more empowered to take a stab at utilizing multiplexing in your own work. So for the agenda today, the topics we want to cover include the benefits of multiplexing. So why multiplex, right? Like you can easily go with single target and not opt for multiplex. And topic 2 would be the factors that impact the success of multiplexing in qPCR. And the third topic would be on the importance of reagent quality and how that can impact your success in developing a qPCR multiplex workflow. And lastly, I will wrap up this presentation with some tips for assay optimization and provide resources for you to go back to at any time if you ever need help with multiplexing in qPCR. All right, so multiplexing in qPCR. So what does that mean? So for qPCR, normally you can have a reaction with your master mix, your sample, and your assay. And when I say assay here, I'm referring to the primer and probes. So one set of primer and probe for one target in a single well on the plate. And so for multiplexing, you are increasing the number of targets. So instead of one set of primer and probe, you're increasing that up to 4 or more, as you wish. And there are other type of multiplexing where you may put 2 different target on the same die and so forth. But generally, this is only done with the TACMan chemistry because in cyber, you're not able to do that because the die will intercalate any double-stranded template. And so say, for example, you have 4 different assay for 4 different targets in your reaction that goes in one well for one sample, and then you can put that on a plate, run it in the instrument, and in the end, in this case, we will have 4 amplification curves for each individual target. And the reporter die difference between the assays are really what allow you to distinguish the different amplification curves. So why bother multiplex, right? You have your workflow already set. Changing it can be a hassle. So to really look into this, here I made up a hypothetical scenario. Say you are a scientist and you have 6, say, gene expression targets that you're interested in studying for a particular disease, maybe, or a mutation. So in a normal single target reaction, you would have to run 6 different reactions in 6 different wells, so each for the individual target. And if you use multiplexing up to 6-plex in this scenario, you're able to run all of that in one well. So instead of 6 wells per sample, you're using 1 well per sample. And so here I'm graphing the total reagent cost. So for reagent here, I included a master mix and assay, and I graph that by the number of samples. So for the navy line, we're looking at the single target workflow, and then for the green line, you're looking at the multiplex workflow. So as you can see that with the increasing number of samples, the cost dramatically increases for the single target workflow. And this really becomes a lot more obvious, right, as the number of samples increase. So this shows us that multiplexing saves costs while increasing sample throughput as well. And in the same scenario, I wanted to look at the number of plates that you need to use as the number of sample increases. So here I'm using a 96 wall plate as an example, and I graph the number of plates needed for the single target workflow in Navy versus the multiplex workflow. And as you can see again, as the number of sample increases, the amount of plates that you use is a lot more. And this tells us that if you change to a multiplex workflow in this case, you'll able to reduce a lot of plastic waste. And that is something definitely that everyone is aware of these days, that we do our best to make sure that we can be kinder to the environment. And just picking, one data point here, you can see if I'm running even just 200 samples, you'll need 3 plates for multiplex workflow versus 11 plates for the single target workflow. And then finally, for this hypothetical scenario to demonstrate the benefit of multiplexing, here I'm graphing the number of hours spent running the plates on a qPCR instrument. So for the single target workflow in Navy, as you can see, the amount of time it takes to run the experiment is a lot longer as you have more and more samples. And again, just looking at one data point, if you look at, say, 280 samples with the multiplex workflow, it only takes 3 hours versus 16 hours for the single target workflow. And for this graph, I haven't even included the amount of time for setup, right? The hands-on time. This is only hands-off time when it's running on the instrument. And as you can imagine, if that time is included, the difference could be even larger. So all in all, these 3 graphs that I've shown for a hypothetical scenario kind of drills down to the point that multiplexing is very useful in that it saves you cost, saves you time, it reduces plastic waste, and also really improves the day-to-day efficiency in lab. So next, I'm going to move into the factors that impact multiplexing now that we've covered why you might want to look into going with multiplexing in your workflow. So the 4 areas that I like to think about for multiplexing in qPCR workflow is the instrument, the assay design, the reagent or master mix, and the thermal cycling protocol. So I will go into more details in each area, but here are some general points about it, right? And we can see that with assay design, there is a lot that you can play around with when you're developing your qPCR multiplex workflow. Okay, so the first step for developing a qPCR workflow, with multiplexing is to check the qPCR instrument that you currently have in your lab, right? So each instrument will have different capability of detecting different dyes. So here on this slide, I have our Thermo Fisher qPCR instrument listed with the dye compatibility listed on the right. So anything that is in green square means that it's compatible. And this information is available online, and it will be provided via link, for by the end of the presentation that you can go back to refer to, and it will have a lot more details on the information, such as whether the instrument comes pre-calibrated with certain dyes, or if you're gonna use dyes that would require custom calibration before you're able to run your experiment with that dye. And another good thing to note is that if you're using a master mix that have passive reference dye, you also want to double check that your instrument is capable of detecting the passive reference dye. Okay, so now that you know the compatibility of your instrument for the dyes that you may want to use, you have to kind of pick what set of dyes will work well together in multiplex, and also the quencher that you're going to pair them with in order to minimize background and limit the crosstalk between the different targets in a multiplex reaction. So for Thermo Fisher, our TACMan probe portfolio offers 6 different dye that can allow you up to 6 targets in a single reaction. So here we have Femvig, Abi, Jun, cyanine 5, and cyanine 5.5. So these are readily available via our custom TACMan probe ordering online, with the exception of Abi and Jun dye when paired with the MGB quencher. So if you're looking for MGB quencher for Abi and Jun dye targets, you would need to order that through specialty oligos in a different section of our website. And something that I really wanted to point out is that the choice of whether MGB or QSY quencher does matter a lot, because it really changes the TM of your probe. So for the same sequence, when used with MGB quencher, the TM will be a lot higher versus using with a QSY quencher. So that is something you could perhaps use to your advantage when you're designing an assay that's specific to your application. Maybe you want a shorter sequence, or if you have to use a longer sequence, you might want to go with the QSY quencher, for example. And so I wanted to make a call out here that the QSY2 probes are new to our offering. So just want to give a little bit more information on that. So these are paired with the cyanine 5 and cyanine 5.5. And we have a great multiplexing application note that is also available online that really demonstrate the multiplexing capability of these probes. And they do have really good sensitivity, good dynamic range, and improved performance when you do multiplexing. And these are all characteristics that are important in multiplexing. And FYI is the turnaround time for these are about 4 to 10 days. Okay, so now that you have your instrument sorted out, and then you have the dye and quenchers that you pick, then it's time to design your assays. So I believe that a lot of us might have some, general rules that they do follow when they design their assay. I mean, I have, similar type of rule, but depending really on the target and application, sometimes you may not be able to stay within the recommended rule, for example, of 40 to 60% GC. Sometimes you might have to stray a little lower or higher because of your application. There's general rule also on having your primer pair within 5 degrees of each other, having your probe sequence TM about 10 degrees higher, and also things like avoiding repeats, and having a good balance of ATGC in the region that you pick for your oligo. And as well as there are avoiding the primer dimer pairs and hairpin structures. So I would say the most important thing is definitely check the specificity for the primer and probes that you're designing. You might already know this, but I want to put it out there that you can use the UCSC in silico PCR to check that. A lot of us use the NCBI blast to make sure that the sequence that you're using are not picking up off targets or pseudogenes. And there are also tools that can predict the interaction between primer and probe pairs. And that we have one also in our website that's called the Multiple Primer Analyzer tool. So that will give you prediction on potential problematic primer or probe pairs. There's also other online tools out there that do that. There's also tools that can predict hairpin structures. But I would say the most, because these are all prediction interaction, right? The most important thing is definitely testing it empirically, and really look out for the complementary basis on the 3' end, because that's what's going to sit down on your sequence first, and then if it gets extended, it gets extended. So that would be something that I also look out for when I design my primers. And since you're trying to do since you're trying to do a multiplex reaction, I would recommend that you start with primers with similar TM values, and similar with probes. Your probes, you want to start them around similar TM values. And that is because if you don't know how they're going to behave, right, and you're using a single thermocycling protocol, ideally you have something that start at similar point, but just know that you might need to adjust it depending on how it looks in first testing in order to achieve the specificity or sensitivity that you're looking for. And I would also recommend having amplicon size that are similar lengths if possible. Again, it can be a little bit different, right? It's not a super black and white thing, but having them relatively similar in length will help kind of ensure that the amplification efficiency will be similar between all the different targets to avoid competition in that sense in terms of amplification efficiency. And I showed you different dyes and in the previous slides. So the dyes are different in their brightness, and you can have FAM-Abby, that's the brightest, versus by Vic and June, and then Cyanine 5 and Cyanine 5.5, that is the dimmest. And there is certain strategy that you should pair your dye with your target. If you know you have a target that's very robust, for example, if it's like a endogenous control gene, like that could probably safely go with the dimmer dye, like Cyanine 5 or Cyanine 5.5. If you have a target of lower abundance, you would ideally pair them with brighter dye to ensure that you can still pick them up in a multiplex scenario. And again, if you are not sure their relative expression level, for example, gene expression targets, there are databases online that allow you to look up, for example, in a specific tissue type, the relative expression of the target. But obviously, if you're short on time, you could always test it out. And if you have your probes ordered with different type of dyes, you can easily swap them, right, as needed, because we don't always know how our targets will behave when we put them in a multiplex, or if we haven't tested them at all before. And the last 3 points is kind of more around optimization after the design. So you can definitely vary the primer probe concentration once you test them, and if you observe some discrepancies, right? Maybe something is showing non-specificity. Say you might want to reduce the primer, or if it's showing higher count than expected, you can reduce the primer concentration, things like that. Or if you suspect that you're putting too much probe that is generating non-specificity, you can also try reducing that, or vice versa. If you have a target that's pretty low in signal, you could also try increasing the probe concentration just to see if it can boost up the signal a little bit. And ultimately, you are using one thermocycling protocol for the reaction. So definitely you can touch that and play around if needed, because sometimes you might have a target that's harder to amplify and require some adjustment. But just know that once you adjust it, right, your other targets, you might fix one target, but your other targets might start behaving differently. So you have to have an open mind going into this, and then adjust as needed. And of course, last point, you might also have to redesign the primer probes and have a different region that you pick instead because of various reasons. So some of you might know already, but I just want to point out that our website have pre-designed tagment assays, and these are for various applications. You can look up the target of interest and in specific species and in specific application, and then they're all shown with links to publications. And we recently actually launched a AI search tool that improved that experience. So this one only applies though for the gene expression and single nucleotide polymorphism assays. So for this new AI search tool, it allows you to make connections between genes and diseases. So you can search via, say you're interested here, we have the non-small cell lung cancer, can search that up, and it can pop up different genes that are associated with that disease based on all the publications that's online. And what this tool is good is that you can also filter them, right? You can filter them by species, by genes, and by tissue type, and then assay type. So a good thing is that you can immediately get a list of the tagman assays with their ID that's linked to this disease that you're looking at, and then you can export that in a easy format to look at in an Excel. And then once you've found your pre-designed assays that you want to put them in multiplex, you would probably want to test them or get multiplexing support first via Assay Design Hub. So that is because the pre-designed assays are currently for single target reactions, right? So you might not know how they're going to behave together, and you may consider submitting a request for bioinformatics help. And of course, the other option is to design your assays, which is totally fine. You can use a design tool of your own choice, or we also have a self-service tool online that leverages our proprietary bioinformatics algorithm to help you design your own custom assay. And again, here you can see that you can choose the tool for the specific applications that you're trying to design your assay for. Okay, so now you have the instrument compatibility sorted out. You pick out your dyes and quenchers, you design your assays, you receive them, and I'm assuming you have a master mix that you you're ready to use to run the multiplex reaction. So this probably will require iterations of runs to optimize depending on how many target you put together. Obviously, the more number of targets in a single reaction will most likely require more optimization. So you definitely want to run the single target reactions for each of the target and the multiplex reaction on the same plate, and ensure that the performance doesn't deviate once you scale up to multiplex, so that for each target, they're looking similar to how they would look in the single target reaction compared to the multiplex reaction. And ideally, you want to test a dilution series of template input that is in your expected range, right? So depending on the sample type, for example, cell-free DNA, it would typically be lower in concentration. So you kind of want to know the expected input. And again, sometimes it might be higher and that it's not an issue, but you might also need to dilute it down. But it's good to have a dilution series of template input that is expected, and then verify that your assay is performing as expected in single target versus multi-target. So some metrics you could look at is that the CQ value doesn't change. With scale up, the assay remains linear, the amplification efficiency is normal, you're not losing sensitivity or LOD, and that there is reproducibility between replicates. Okay, so now you've run iterations of your assay, changing concentration, whatnot, and supposedly now you got to your optimized assay. So here I'm trying to show you what you should expect when you do a multiplex, when you have an optimized multiplex assay. So here I have a 5-target reaction ran with individual target for each of the 5 target in that multiplex reaction side by side on the same plate, and I'm plotting the delta CQ, so the CQ difference between single target versus multi-target performance. And here just for curiosity, we wanted to see like, oh, okay, the same assay, we have 2 instruments, does it run okay in a different instrument as well? And as you can see, that seems to be the case for both instruments tested here. We have CQ value that remain similar with scale up to multiplex. So they're all within 0.5 or less difference, which is awesome. And I want to really emphasize that at Thermo Fisher, we have good quality system to and audits, regular audits, and things that control our facilities that manufacture these TACMan assays, so that we can ensure that they are consistent in performance. And every time you order a made-to-order assay, you will be provided a CFA. And our pre-designed assay are guaranteed for performance for single target reaction, and they're verified with up-to-date annotations. And then for more terms and conditions of this guarantee, you can look that up online. So really wanted to talk about next is quality, right? So earlier I have shown you kind of the factors that impact multiplexing, and then the general guideline of how to go about optimizing your assay a little bit, and then getting to that point where you have a optimized assay. So obviously you will use a master mix for your multiplex reaction, and that maybe you haven't thought about too much about it, but that does also matter, right? So why does that matter? So first of all, I'm just gonna show you the portfolio of qPCR master mixes that our company provides. And today specifically, I want to talk about Duraplex because that's our latest master mix that went out on market, which allows for higher order multiplexing. So we have two versions of it with passive reference that you can amplify up to 5 targets, and the no passive reference allow you to amplify up to 6 targets. And then this is a one-step master mix that uses RNA as input. So this master mix, aside from its multiplexing capability, it actually have a lot of great features that I want to just briefly mention in case that is specific to your application needs. So it has really good stability. It has lower viscosity that allow you to test in liquid handler system if you want. We have a great stability white paper on this master mix on our website as well if you're curious to look at it. And it also have things like good inhibitor tolerance to common inhibitors found in clinical samples. And of course, it's manufactured in a regulated facility, and each lot is tested as it comes out to ensure that we're providing something that is of good quality. Okay, so in an ideal optimized multiplexing workflow, so I've mentioned you want to test, when you're optimizing, you want to test a dilution series of template inputs. So here we have a 5-target reaction amplified using Duraplex. So I have 2 different inputs. So the dye filter 1, 2, 4, and 5, they are from the same target, so RNA, or same template, sorry, RNA. And then the line that's going straight across that have same CQ across is not diluted. So I kept that template input constant. I believe that's an IPC that we used there. So here, this testing is to kind of see if there is interference of amplification of a very high copy target on something that's lower concentration. So I kept the IPC at a pretty low input, and the RNA for the 4 other targets ranges from low to high, but you can see that there is no interference at all. The assay performed linearly for each of the targets. And importantly, the target that's kept at 1 input remained constant throughout the different different concentrations of the other template input. And just picking, one of the data point here and then graph it out in terms of the CQ difference between the single target reaction, which was also ran on a plate, compared to the multiplex reaction that I'm showing on the left. You can see that the delta CQ again is less than 0.5 difference. So showing that with scale up, there's no loss in performance. So you might wonder what things will look like if you actually have competitive inhibition between your targets. So here's just some graphics of, say, 2 targets in a single reaction. So I have target 1 in green, target 2 in navy, and target 1, I'm doing that linear dilution, zero dilution of template input, and then target 2, I'm keeping the input at a fixed concentration. So in an ideal scenario, you observe what you see in the middle, right? So it's similar to what we see in the previous slide, but when you have a scenario where things are, when there's competitive inhibition between the targets, you may see that these target 2 here in navy is having delayed CQ as you have a high concentration of target 1. So something you might do here, you could probably reduce the primer probe concentration for target 1, right? And then see how that looks. And so Duraplex, Duraplex is made for multiplexing, and it has a very wide dynamic range. And you might ask, why is that important? So we want a master mix that has wide dynamic range capability because we want to ensure that you're able to amplify all of the targets that you're looking at in a single reaction, even at lower copy numbers. Because sometimes with scale up to multiplex, you can lose sensitivities. So here I'm displaying a 6-target reaction using Duraplex as the master mix. And I have a zero dilution of 9 different concentrations. And as you can see here, that all 6 different targets amplified, beautifully, consistently with similar spacing between the zero dilution. So with wide dynamic range, you will have less worry about losing the amplification of certain targets with scale up. And to really kind of drill that point down, right? You could say, okay, so what would it look like if we actually use a master mix that is not designed for higher order multiplexing? So here I ran the same assay, 6-target human gene expression targets with either Duraplex master mix or a supplier 2 master mix that is advertised for up to 3 targets. And I know that's not a fair apples to apples comparison, but here we're just curious about what happens if you actually use something that has lower multiplexing capability. And as you can see on the top, the 7 dilution points of the template input was able to be amplified linearly. So I have the R-square values plotted up there too. Sorry, this is the CQ by log RNA input plot. And on the bottom, the supplier 2, you can see that you are only picking up anywhere from 2 to 4 dilution point, depending on the target you're looking at. And furthermore, the linearity of the assay is also reduced, right? You can see the R-square value is not quite as close to 1 as you see on the top. And then with the gray areas that's shaded around the line of data, that is the confidence interval, and you can see that the consistency is reduced also with a master mix that is less capable for multiplexing. And also, I just want to make a note that it's only picking up fewer dilution point, which also indicates that there is a loss of sensitivity. And finally, for Duraplex, I wanted to demonstrate another metric that you may look at, right, when you're optimizing your multiplex workflow is reproducibility and consistency. So between your runs or replicates. So here we have Duraplex with no passive reference on the left, and the version of the master mix with rocks in it. So we're testing either 6 targets on the left versus 5 targets. And we have 3 independently manufactured lots of the master mix tested side by side, and you can see that the 3 lots of master mix performed very consistently across all the targets that are looked at in the multiplex reaction. So all in all, I just want to really emphasize that it is important to have good quality reagents so that it will make your optimization process a lot less painful, and ensure that you can get the accuracy, the sensitivity, and then the reliability of the results that you are getting. And so all our TaqPath master mixes are manufactured in facilities that adhere to the CGMP principle and is ISO 13485 certified. And then the TACPath Master Mix portfolio also have the compliance documentation package to help support regulatory submission if you choose to use one of them for your workflow. Then finally, in the last bit of this webinar, I want to go over quick tips around optimization. I kind of talked a little bit about it in the assay design portion. I also mentioned it here and there throughout the presentation, but I want to kind of wrap up this webinar by reminding you what are different things that you can change to optimize your multiplex workflow. And in the end, I will also provide resources that you can go back to in developing a multiplex qPCR workflow. Okay, so as we have shown in the beginning of the talk, we know that multiplexing is beneficial, right? It saves time, it saves money, it reduces plastic waste, and it improves efficiency. But as we move on to the next section of the talk, right, we talk about the many different things that you need to look out for when you're developing the multiplex workflow. And then specifically the assay portion of it is, kind of scary, right? It's a lot of things going on. Maybe not scary for everyone, but if you're not used to it, it can feel intimidating to want to try. But I want to help you look at it this way, right? So there's actually a lot of different knobs you can turn when you optimize your assay, and that means that you have the freedom to make things to optimize things to the point that is good, right? So so think of it as not limiting. There's a lot of things that you can play around with, but you just have to know you have that freedom. And maybe if you're short on timeline, you might also want to have more oligos ordered so that you don't have to wait for them and they're ready to swap out. So for example, for a sequence location in your assay design, I said you want to start with similar length, but sometimes that's just not feasible, right? You might have a region that have a lot of repeats, or you have region with stretch of G and C, so then which can make things difficult. So you might have to move your sequence around, or you also need to check, say if you have pseudogenes that you don't want to pick up, you may want to strategically position your primer and probes in regions that are mismatched between the gene of interest and the pseudogene to ensure you're not getting off-target effects. For reported dye and target pairing, I talk about it, but just as a reminder, you want to pair brighter dye with something that is less robust or of lower abundance. And again, you might or might not know that. And sometimes you just need to test it out to see. But again, you have the option to have probes in different dyes so that you can swap them out when needed. So again, frame Ab as the brightest, followed by Victune, and then cyanine 5 and cyanine 5.5. And other things you can play around with is like the concentration of primers. If you have something that's a little bit harder to amplify compared to the other targets in a reaction, you might consider increasing the concentration. Or if you're seeing non-specific reactions, you could potentially also reduce the concentration of primer or probe. So probe concentration definitely can play around with if needed. Obviously, you start with maybe similar concentration for all of them in the beginning. Unless you already know something that's going to be really robust, you can start that with a lower concentration compared to your other targets. And if you see non-specificity in your probe, you may consider reducing the concentration. You may consider reducing the TM of your probe so that it is more restrictive with the same thermal cycling protocol that you're using. And same thing with the TM primers. You can play around with that. So what you can do is when you're ordering things, you might have stuff that are couple base pair difference with different TMs ready to go, so that when needed, it's easy to swap them out and test. And lastly, the thermal cycling protocol, definitely something you can play around with. There's the denaturation temperature that typically is enough for most target, but the time you may change if it's something that you know that needs a little bit longer time to open up. And then for the anneal extend temperature, that is something that is played around a lot more, depending on your primer probe TMs, right? But just know that when you adjust the thermal cycling protocol, you might improve the performance of one target, but end up changing how the other targets might behave. So really having different versions of the primers and probes can help reduce the amount of time it's needed to develop a qPCR multiplex workflow. And then we have survey support and tools to help you develop the workflow. We have a very nice qPCR multiplexing learning center hub online, and there's a lot of different resources. There's a PDF that goes over like different steps of optimization. You want to different steps of optimization for different type of assays, applications, and also specific guidance around assays for different application. The instrument graph compatibility die, the instrument to die compatibility graph that I showed earlier in the presentation is also can be found in that hub. Some things I mentioned earlier, the multiple primer analyzer tool is here. There's the pre-designed assay search tool, the assay design hub. And I believe if you're registered for this talk, you'll receive the links to this. And I have, and the last thing that I haven't talked about is the OEM solutions. So this is custom commercial solutions. If you're looking to outsource a specific part of the assay workflow development, we also provide that service if needed. Okay, so finally, I want to touch upon our company's internal goal of sustainability. So our current goal as a company is to achieve net zero scope 1, 2, 3 carbon emissions by 2050. And this is something that I think is great because like, you know, during this time, we're very aware of the impact we have on the environment. And as scientists, that can be hard, right? We use a lot of plastics and whatnot. So really be conscious of how you're disposing your waste. If there are ways to improve shipping methods so that it's more environmentally friendly, definitely do that. So we're constantly doing that, especially, you know, I work at the South SF site. We have great waste streams that make sure things are going to a better waste stream if they are able to. And so things are sorted out very well. And we actually just received the highest Green Lab certification last year. So yeah, very happy about that. But yeah, just want to share that we as a company, we do care about that too. And with that, I want to thank you all for your attention. And I hope that today you learned something or you enjoyed the talk, in that there's something that you can use in the future, whether it's a tip of advice or some type of resource. And yeah, I'm happy to take any questions.
All right. Thank you very much for that great presentation. We'll jump into the Q&A now. So if you haven't sent in a question, do so now. We'll try to get through as many as we have time for. So the first one I've got here is, can you tell me a bit more about MGB versus QSY for assay design?
Yes, so the MGB and QSY are the different quencher types for different probes. So the MGB really increases the TM, right? So you can use that to your advantage. So sometimes you want a shorter sequence. So the MGB will allow you to have a shorter sequence, and it can improve specificity by increasing the TM. And yeah, if you have any more question around that, we have resources online, and also feel free to submit more questions around it in the chat. All right.
The next one we have here is, are there multiplexing capabilities for combining Hackman assays and assays from IDT, for example?
So that I don't have any data on that. So yeah,
Okay.
we'll have to move on to the next question.
Okay. Let's see here. The next one we've got
Sorry,
is it.
Sorry, just want to add on to that. If you want to do that, you will have to, again, make sure your instrument is capable of the different dyes that you're putting together from the two different companies, and that they're properly calibrated. And you might just need to have to test it out to see. Thank you.
Okay. Are there multiplexing? Oh, nope. Next one here. Is there a good tool or site that Thermo Fisher has to predict pseudogenes?
So not that I'm directly aware of what's on our website. So for me specifically, I use NCBI Blast a lot, and you can also just have a software that you can pull the sequence of your gene of interest and the pseudogene pretty easily from databases like UCSC or NCBI, right? And if you have a software that can align them, which I believe there's also free software like APE, that you're able to align the sequence to make sure that your primer and probe, at least one, is sitting with enough mismatches with the pseudogene. And ideally, you want the mismatch closer to the 3' end, right? Because we know that's where primers sit down. So if the mismatch between your gene of interest and pseudogene is closer to 3' end, or if it's an insertion deletion, you might want to strategically put it there to make sure you're not picking up the pseudogene.
All right. Next one we have is, does Thermo have designs already designed for target and normalizing genes?
For target and yes, so that we have a big portfolio of the pre-designed assays. So they're specific to different application types. So if you look in our website, Tagman assays, and that should take you to the search tool where you can specify the species you're using and the gene of interest, and then look into there. And we do have normalizing genes like RNase-P, GAPDH, actin, a lot of them.
All right. Thank you. Are primer and probe sequences provided for pre-designed assays?
So unfortunately, that is not provided. So if you want the sequence, you would have to go through the self-service assay design tool.
All right. Let's see here. Which master mix is best for two targets, a gene and normalizer gene?
So that I'm gonna make a joke here. That feels like a trick question, but it depends on the context, right? So you'll have to look at the application, your sample type, because if some sample types are a lot easier to or cleaner to amplify from, so then you won't care about the inhibitor tolerance capability of the master mix. So you can look for something that doesn't specifically advertise for that, but for sure you would want something that can claim at least up to 2 targets, which I believe we have a lot. Most of our master mix are capable of doing 2 targets.
Are there any product designs information for multiplexing in dPCR?
That is a good question. I can follow up with you on that if you want to submit. I think we have, remind me, Matt, we have a way for people to submit. Okay, we can follow
Yep.
up and send you information with that. I can't remember off the top of my head what type of information we have for the dPCR part, but I believe we have something. But I can get back to you on that.
Yep, we'll pass that along. What is the purpose of passive dye, and is ROX a passive dye?
Oh, yes, I apologize for not explaining that ROX is actually a passive reference dye. So that dye comes up in the June channel typically, and a passive reference is a good thing to have to control for the variability in pipetting between different wells.
All right.
It's like a normalizer so that for the other targets, so that if you have the variation in your pipetting, it can normalize based on the amount of passive reference in the reaction.
Okay. We've got time for maybe 1 or 2 more.
Okay.
Let's see here. Would you recommend multiplexing PCR to detect changes in expression of different splice variants of the same target gene? And are there ways to improve detection of low abundance targets?
That is a good question. So if I can imagine you probably want to do that anyway if you're trying to detect different splice variants within the same sample. So that can be done for sure, just depends on the splice variants, like how different are the sequence. So they will probably have different introns splice out. So you can definitely utilize that to your advantage and design something that will sit in the exon to exon junctions. So you have to look at between the variants, like, so all of them have these exons, these introns spliced out, but some of them will have maybe exon 1 and 5. I'm just making up things, 1 and 5. It could be 1 and 3 that's combined. So then you will design a sequence that's in between a junction to ensure that you can detect the different splice variants. But yeah, I think that's doable. And the ways to improve detection signal is one of it is you can try increasing concentration of primary and probe. And then assuming that the primer sequence themselves are robust enough in picking up the target.
All right. Well, thank you very much again for your great presentation and for your time and all the important research. I'd like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's webcast. I would like to thank the audience for joining us and all the questions. Questions we didn't have time for, and those that do get submitted during the on-demand will be submitted to our speakers. We will have this on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who might have missed today's live event. So until next time, thank you very much once again, and goodbye.
Thank you. Goodbye, everyone.